So the venous system has really taken second place to the arterial system for a very long time. But if one looks back historically, we can see something very interesting. At the beginning, we really didn't even understand that blood flowed throughout the body in a circle, that the heart pumped it. It was really William Harvey who came along and he realized that veins had valves in them. When he understood that, he realized, okay, what are the valves there? They're stopping the blood from flowing backwards along that direction. And it was with this simple concept that he actually was the first one who said, aha, the blood must pump in a circulatory fashion throughout the body. So veins already played a very critical part of that research. And now with Zamboni's work, we're beginning to rediscover the importance of the veins. And another comment I would just generally make is, when we look at the veins in the brain with some of the special technology I'll show you today, we can see the effects of changes in oxyhemoglobin. So when the arterial blood comes in, it's mostly oxygenated blood. When venous blood comes out, about 30% of that oxygenated blood is now deoxygenated. Not all of it, just about 30% of it. But it's that presence of deoxyhemoglobin that tells us how well the brain is functioning. If that blood came out 100% oxygenated, you would know that that part of the gray matter, or that part of the brain tissue, was not working properly because it wasn't taking out the oxygen. So in fact, the venous side of the system tells us an incredible amount of information. It tells us how that oxygen is used. Now with MR, we can get beautiful images like this showing you the blood vessels in the brain. Actually, this is a three-dimensional information that we then just project onto a 2D image. The doctors can look at this and say, okay, do you have narrowing of your vessels? And this is just for the arteries in this case. We can also, with these technologies, look at the veins in the same way. The image on the right-hand side is from a cadaver brain where they injected a dye into this, and then they did a radiographic image. Beautiful. This is probably one of my students, and I'd like to tell you that I don't sacrifice my students to validate this sort of thing. So we're very careful about that today, you know, all these human study approvals and things that we need. And so you can see that this cadaver brain example shows very nicely that what we think we see are veins in this type of technology, they really are veins. Now, just to go back a little bit historically, and I hope you'll forgive me, because if I talk at this pace, I'm going to be about 2.5 hours, so I'm going to have to skip a few slides. So I'm going to have to skip a few slides here in order to stay on time, so I don't get the Scottish hook. And let you know that these slides will be available either through Tom on a website or on our own website, ms-mri.com, so you'd be able to go and look at more details of this. But just to point out to you that even back in 1839, people recognized there was abnormal vasculature associated with multiple sclerosis. Perhaps some of the most important work was by Putnam in 1935, because Putnam already realized that it was probably obstructed venous flow that was the most important driving factor behind MS. In fact, if Putnam had had an MR scanner in his day, he would have already discovered CCSVI, because he believed from 1935 until his death, which was 30 or 40 years later, he believed it was obstruction of the veins that caused this problem. So other people after him, they also did studies on cadaver brains, and they found the same type of relationship between the venous system and the lesions. Fogg was one, Franz Schelling, who was here yesterday and may be here today, I'm not sure. He was also a strong proponent of the role that the veins played. And then, of course, now Zamboni in 2009. But I always enjoy this particular slide because Tracy Putnam was a neurologist and an excellent experimentalist. And he did a study on 14 dogs, and he demonstrated that if he obstructed the veins of these dogs, that they developed sclerotic lesions, very similar to what happens in multiple sclerosis. So his quote at the end of his paper, this is a verbatim quote, is the similarity between such lesions and many of those seen in cases of multiple sclerosis in men is so striking that the conclusion appears almost inevitable that venular obstruction is the essential immediate antecedent to the formation of typical sclerotic plaques. So that type of evidence has been around for quite some time. And then, of course, we have Zamboni, who noticed instead of studying the brain like Putnam was doing, he looked outside the box. And outside the box, he found problems in the major veins, the jugular veins, the azygos vein, and I would suggest that, in fact, there are probably lots of other problems here as well, not just those veins. And the more we look at this from an imaging point of view, whether that's the venography or the MR, the more we're going to discover. I mentioned a little bit briefly about vitamin D, but in a slightly different way than people have talked about it before. There is some work by Sissek and Stein that point out that vitamin D deficiency also has implications for hypertension and atherosclerosis. And also, Nemirovsky shows the same thing. Vitamin D deficiency is implicated in vascular diseases. So vitamin D may play some role also with the endothelial health or the vessel wall health. And it may be more than just having an importance with respect to bone and teeth and things like this. It may be important for the vascular system. Now, reduced perfusion is an important part of this, and I'm sorry this is a very busy slide, so again I'll just summarize this. But more recently, people have been using MRI to study the cerebral blood flow. How much blood gets into the tissue and how quickly does it get there, basically. And they have found that in MS patients, if you go from normals with fairly high blood flow to CIS or first symptom patients, the blood flow goes down a little bit. Then you go to relapsing remitting MS patients and the blood flow goes down more, and you go to primary progressive and it's down even further. So in fact, something has been affecting this. And this work in the MR imaging was just done in the last 10 years. Again, it was pre-Zamboni, so there were a lot of hints there that there's something wrong to perfusion to the brain. Now, of course, the question is, was that caused by something else? I mean, the neurologists are going to fairly ask, you know, were there other neurological problems that then led to vasoconstriction, for example? That's certainly possible. But reduced perfusion also led to a fellow by the name of Gerling to look at this and say that maybe there's a role of hypertension associated with this. And you'll notice in brackets here I say, recall that reduced vitamin D can also lead to hypertension. Now there was an interesting study that was done on a large number of elderly people who had hypertension. And they found out that a lot of them had vitamin D deficiency. When they got increased amounts of vitamin D, not all of them, but a certain fraction of them no longer had hypertension. So some interaction was going on there to improve perfusion. So there was a lot we don't know yet. And some of these tools that we're looking at are telling us there are abnormalities. We may not understand those abnormalities yet, but our goal, of course, is to understand this. Reduced perfusion can also lead to hypoxic problems. So you're not getting enough oxygen to the tissue. And Lucinetti noticed in a different form of MS, Balo MS, that there were these hypoxia-like tissue damages taking place in the baloconcentric rings. So all of this is hinting to bad perfusion, bad delivery of oxygen to the tissue. So, speaking of which, I shouldn't be doing this. So you might have noticed I started this at the beginning of the talk. Coffee's not good for you. 2 cups of coffee can have a major effect on the blood flow to the brain. Coffee is a vasoconstrictive agent. But this is kind of interesting because you could use something like coffee as a means to study the cerebrovascular reserve. How well does some person's brain respond to a challenge? If you're healthy, you'll respond to that challenge fairly well. So it's a pretty harmless contrast agent. So here are 2 images. The one on the left is a gentleman that has had no coffee yet, and he's not a coffee drinker. And the image on the right shows what happens after he's had 2 cups of coffee. This is about half an hour later. And you can see that here, these small veins don't show quite as well, but afterwards they show very well. In fact, if you look at this, you can see these vessels are much darker here. And the reason is that because the blood flow is slower and the brain is still working at the same rate that it did before, you have much higher levels of deoxyhemoglobin present in those veins. So this person responded quite well. And one would think then that if you had reduced blood flow, you would see these veins much better. Well, you're going to see later on some results by Yu and Ge, and also more recently, Zavatinov has replicated something like this, where we see the very opposite for MS patients. So one has to ask why in that case.